Hi, everyone. Can everyone hear me? Hello? Okay. So today I will be presenting our work on ChIP-seq signal normalization. And so to start with, I'll just go over a really brief overview of ChIP-seq. So in short, what it measures is protein activity along the genome at different positions. And so the activity that you're usually measuring is either transcription factor binding or histone modifications. And so if you imagine a really simplified genome, like what's shown here on the left, with a single bound protein in red, then running a ChIP-seq experiment will give you something like the histogram that you observe here, where you have the highest signal at the actual position of binding, and then it kind of tapers off towards the ends. And you often have a bit of noise towards even regions where there's no real activity going on. Now that we have a way to quantify this kind of protein activity, we can go ahead and run some quantitative analyses to find patterns in transcription factor binding and histone modifications. But when you're searching for these broad patterns and don't know exactly what you're looking for, you will typically want a fairly large number of experiments, which can be tricky in this case because these experiments are very expensive to run. And so the good news is that there are multiple large-scale projects which have ran essentially hundreds of these experiments and open sourced their data for computational biologists to use. And some of the more major ones are ENCODE, IHEC, and Roadmap Epigenomics. But how do we actually go ahead and compare these experiments quantitatively? Well, we can consider two examples. So on the top, we have a section of a transcription factor binding ChIP-seq experiment. And on the bottom, we have a histone modification experiment. So starting with the top one, we can see that in general, signals are pretty low, with the exception of these narrow peaky regions where the transcription factor binding actually occurs. And when you're working with these kinds of data sets, what people typically do is they just use the called peaks and pretty much ignore everything else in their downstream analyses. And there's been a lot of work done on accurate peak calling methods, and they tend to work quite well. But if you look at the bottom experiment, things actually become a good bit more complicated because you can see that the signal is much more uniform. And so if you only look at the regions which appear to be peakier than their surroundings, which would be here, you would still lose a lot of information if you just throw away everything else. And so when you're working with these kinds of data sets, what you typically want to do is look at a much larger portion of the genome. And there's no general consensus on just how to use this arbitrarily for any use case. But what people typically do is you take these signals, feed them into some machine learning model, which is trained to perform the task that you're interested in. And usually there's some kind of domain-specific pre-processing that you need to do. Okay, so now that we have a large number of these experiments and we know how to quantitatively compare them, we can go ahead and use them to detect more differences in the epigenomes of different cell types and samples. But it actually turns out that just doing a very naive kind of apples-to-apples comparison of the signals that come out of these experiments is not a very good way to go about doing your analyses. And the reason is that there's a very large number of technical noise factors, which can basically mask the real signal that you're looking for. And so what can happen is often you will see that different experiments that were produced by different labs will have very different distributions. And the reason can be just the different ChIP-seq protocol that they used, maybe different antibodies, different reagents. And so what you want to do in this case is to use a normalization method, which will essentially reduce all of the technical noise and then hopefully maintain as much of the biological signal as possible. And as a really brief example, if you're interested in comparing these three samples, if you just look at the signal intensities, then you would think that sample 3 has the most biological activity going on. But what could also be going on is that the technical noise is what's causing all the variance between the different experiments. And this actually ends up being the case here because these regions essentially focus on a subset of the genome which exhibits pretty uniform activity throughout and across different cell types. And so everything that you're observing here is just the result of technical noise. So one really prominent real-world example of why this type of normalization is needed is the ENCODE imputation challenge. So this is a challenge which was run by the ENCODE consortium, where people or research groups were asked to create ChIP-seq imputation methods. And so these are methods that take observed conducted ChIP-seq experiments and then try to predict what the held out signals from unconducted or unobserved experiments would look like. And so the matrix on the bottom left here kind of illustrates the setup. So on the x-axis, we have different cell types. On the y-axis, we have the different targets. And then the task here is to take the blue and orange experiments and predict the green ones from them. And the matrix is so sparse because, again, these experiments are very expensive to run. And so if you can do kind of computational imputation reliably, then you can save a lot of time and money by filling in this matrix. But what actually ended up happening is because of big distributional shifts between the training and the testing data sets, there was pretty much universally poor performance across all submitted methods, even though they were doing well on the validation sets. And actually, pretty much almost every method ended up doing worse than a simple average baseline. And again, the reason is this bias, which was later corrected. And as another quick example, we have segmentation and genome annotation algorithms, or SAGA, which take a set of ChIP-seq experiments as input, and then they output position-based or location-based clusterings of the genomes and potential functional labels, such as promoter or enhancer. And so one approach to using SAGA on multiple experiments at the same time is to concatenate the ChIP-seq tracks, which are produced by these experiments. And what this ends up doing is it forces the model to learn essentially common patterns across the different experiments. But when you just, again, naively concatenate things and run SAGA on them, then the model will potentially be confused because you will have these experiments which have very different underlying signals. And so again, this is a domain where it's pretty important to apply normalization properly. So hopefully I've now somewhat convinced you that some form of normalization is needed when working with this type of data. And I'll just briefly go over some existing normalization methods which are commonly used in practice. So the very first one, or the first two, are the square root and log transformations. And so these are more technically variance stabilizing transformations, but they do end up working pretty well as normalization methods. We also have something like quantile normalization, where you pick one of your experiments as a reference, and then you take the signal distribution and force all other signal distributions to match it. We have fold enrichment, where essentially you incorporate input control signals by taking the ratio of your actual experimental signal over the control, and you typically want to add some kind of pseudo values so you're not dividing by 0. We also have Poisson p-values, where you fit a Poisson model to the input control sequence and then use it to normalize your experiment. And then finally, we have something like S3 norm, where you look at common peak and common background regions across all of the experiments that you're comparing, and then you learn a exponential transformation, which forces the mean signals within these common regions to be the same. And so now I'll go on to explain some of the intuition behind our method. So because ChIP-seq has inherent randomness in the way that it's designed, we model the number of reads that occur at each position in each experiment probabilistically. And so the idea is to somehow learn a distribution of the background signal, which can be explained by technical factors. And then to get your normalized signal, you just look at where your actual observed reads falls along that predicted distribution. And so importantly, most or many existing methods rely on training models that are specific to the experiment or the set of experiments that you're interested in comparing. And what we do here instead is we train our model on whatever experiments we have available at the time, and then you can apply it to new experiments without having to do much to normalize them. And so the schematic in this slide and in the next few slides outlines the steps of the normalization approach that I just described. And so if we think about a set of experiments that we're interested in comparing, each experiment has its own ChIP-seq read counts, and then it has associated covariates, like the antibodies that were used for the experiment, the sequencing machine, the lab, and so forth. And so what we can do, or you can select basically any of these covariates that you would be interested in, but the important thing to consider is that they should not really leak any of the information about the biology that you're interested in measuring, and they should just be essentially potential sources of technical noise. So once you have decided on the set of covariates to use, you can go ahead and feed them into a simple, fully connected neural network, which was trained to predict the parameters of a negative binomial distribution. And so what this ends up producing is a position-specific distribution of expected read counts. So the next thing you do is you can take the observed number of reads in the experiment and then calculate their quantiles within the predicted distributions. And so in general, the quantiles will be pretty high for regions where there are a lot of reads, and they will be on the lower end in background regions. So now at the end of this step, you've gone from read counts from the original experiment to quantiles within the predicted distributions. And then in the final step, what you can do is you take the quantiles within the predicted distributions and then use them to apply quantile normalization to some reference distribution. And so this reference distribution can either be a theoretical distribution, such as an exponential distribution, or it can be an empirical distribution, where you just pick one of your samples underlying signals as the reference. And at the end, you get these normalized signals that you can hopefully compare more directly, because they are now, by definition, drawn from the same distribution. So I'll only have time to go over one set of results, and it involves using ChIP-seq signals to predict gene expression levels in the different experiments. And so the idea here is we use this kind of surrogate evaluation to assess how well our normalization is going, because it's really difficult to measure that directly because you don't have access to the ground truth. And so instead you rely on some other kind of predictive tasks to see how well you're doing. And so the matrix on the left here gives an overview of the evaluation. And what we can see is that different ChIP-seq experiments are on the x-axis, and on the y-axis we have different genes in the human genome. And then we divide it into 3 regions where we train on a subset, we train a linear regression model on a subset of the genes and a subset of the experiments to predict gene expression. And so the input to the linear regression model is a set of 3 features per genes per experiment. And so the first feature is the mean signal in a 2 kb region centered on the transcription start site. We also have the mean signal in a 2 kb region centered on the transcription termination site. And then finally, the mean signal in the remaining gene body. And we take these 3 features per gene per experiment, and we train a linear regression model on the blue region, and then do 2 separate evaluations on the green and orange regions. And we do, and we repeat this in cross-validation splits. Okay, so on to some results. This plot shows the results on the held out genes in the cross-validation splits. And so from the previous slide, this would be the green rows that the model was not trained on. And so as a reminder, this is training the linear regression model on a subset of the genes, and then using it to predict expression in genes that it's not seen before. And the general idea here is that if the signals were normalized well, then they would have washed away any source of technical noise. And then the remaining true signal would allow a simple model like linear regression to make better predictions. So on the x-axis, we have the different assays that we're looking at, and then on the y-axis, we have the difference in model R squared between the normalized and the unnormalized signals. And so, for example, what we can see here in the first column is that all of the normalized signals outperform the unnormalized ones at the predictive task because everything lies above y equals 0. And then we can also see that our signals outperform the other normalization approaches that we tested. And then the pattern holds generally for other assays as well. So for most assays, all normalization signals tend to do better than unnormalized signals. And then our method performs comparably to or better than other normalization methods, except for K36ME3. And we're developing the model a bit and hoping to bring these numbers up. And then very quickly, this is the other evaluation which I mentioned. So this would correspond to the orange column in the matrix from a couple of slides ago. And again, the pattern is pretty much broadly the same, except that the absolute differences are smaller, both between unnormalized and normalized signals and between the different assay types, normalization methods. Okay, so to wrap things up really quickly, the takeaways here are that we can use quantitative comparison of ChIP-seq experiments to study different patterns of transcription factor binding and histone modifications and just better understand patterns within the epigenome. But it's usually ineffective to use these unnormalized signals as they are because there's big distributional shifts and sources of technical variance, which make direct comparisons difficult. So in this project, we develop a normalization method which is motivated by the inherent randomness and stochasticity of ChIP-seq experiments. And hopefully we can use these well-normalized signals more in downstream applications to basically learn better biology in applications such as imputation and SAGA. Thank you.
Thank you, Abdul. Do we have any questions? Questions online in the chat?
There was one late question for her, but I don't know if it's...
Do we want to do that right now? All right, let's see if any questions for Abdul, or was the presentation completely crystal clear? Yeah? Okay. Oh.
So are you going to use this for data in your lab only, or are you going to go take public sets and redo everything?
Oh, yes. So to clarify, we're using data from the ENCODE consortium.
In the encode. So you're going to but are you going to reprocess the encode with this, or with this new approach?
We're hoping to essentially train. Basically, you can compress the information into a small model, which you can then use to train or to normalize new experiments.
Okay, so the question to Beatriz is from, hello, I'm Emmanuel Martorana from the Mediterranean Institute of Oncology. I would like to ask you what the procedure is with patients when there is an update, like in your example, the addition of a drug. The addition of a drug.
Do we know which presenter that question is for?
It was in the end of Beatrice's talk. Yeah. So, I would like to ask you what the procedure is with patients when there is an update, like in your example, the addition of a drug.
Yeah, so I don't think we update any of our older clinical reports. So for any of the clinical reports that were already produced, there wouldn't be any information back to them. But for future patients, that would show up in the clinical report. So I think if that makes sense.
Thank you.